Welcome back, class. In today's lecture, we're going to explore an important part of the field of microbial ecology. And we're going to do this by discussing the process of elemental cycling. That is the exchange of nutrients by microorganisms that makes life on Earth possible. This is sometimes referred to as environmental microbiology. We're also going to look at some of the more important tools and techniques that are used to study microbial ecology. Moving on now to the nitrogen cycle. And just like the carbon cycle, the cycling of nitrogen makes life on Earth possible. Here we have a simplified nitrogen cycle. And I'd like you to note the different arrows presented here. Let's look at what they represent. So you'll see to the right of this cycle two white arrows, and these represent flows that occur mostly under aerobic conditions. Then we've got a couple of solid bold arrows, or just plain purple. These arrows represent anaerobic processes. You can also see some cross-barred arrows, that is blue and pink arrows. These represent processes that occur under both aerobic and anaerobic conditions. Also shown here are some of the important genera that contribute to different components of the nitrogen cycle. So genera including Pseudomonas, Azotobacter, Clostridium, Nitrobacter, Nitrococcus, Solvovibrio, Geobacter, Nitrosomonas, and so on. Here you can see that I've broken down the nitrogen cycle into smaller components, so it's easier to follow. And we're going to begin our discussion of this cycle with the fixation of the inorganic or molecular nitrogen, N2, into its organic form, that is ammonium ions or amino acids. Nitrogen fixation is uniquely a prokaryotic process, and apart from a limited amount of nitrogen fixation that occurs during lightning strikes, all organic nitrogen is of prokaryotic origin. Nitrogen fixation can be carried out under both oxic and anoxic conditions, as represented by these coloured arrows. Microbes such as Azotobacter and certain cyanobacteria can fix nitrogen aerobically, while free-living anaerobes such as members of the genus Clostridium fix nitrogen anaerobically. But of course, the best studied nitrogen fixing microbes are the bacterial symbionts of plants known as legumes. And these bacteria include members of the genus Rhizobium. The first product of nitrogen fixation is ammonia, NH3. And that's not shown here, probably because it's immediately incorporated into organic matter as an amine. You might remember the amines from your organic chemistry in both Living Systems 2 and wine chemistry. Now, the addition of electrons for every nitrogen requires lots of energy and a fair amount of reducing power. So the nitrogenase enzyme that catalyses this reaction is very sensitive to oxygen and has to be protected from oxidizing conditions. Now, ammonia made by nitrogen fixation is immediately incorporated into organic matter as amines, as I said, and then these amine nitrogens are eventually introduced into proteins, nucleic acids, and other biological molecules. The nitrogen cycle then continues with the degradation of these molecules into ammonium ions, which you can see at the bottom of this cycle, NH4+. Now, one important fate of the ammonium ion is its conversion to nitrate, NO3-. And that's not quite shown yet, but this is a process called nitrification. It's a two-step process whereby the ammonium ion is first oxidized to nitrite, which is shown here, NO2-. Just follow that white arrow. And then the nitrite is oxidized to nitrate. Bacteria of the genera Nitrosomonas and Nitrosococcus, for example, play important roles in this first step that's shown. Here we have the second part of the nitrification step, oxidation of nitrite to nitrate. You can see nitrate, NO3- at the top of the cycle. Nitrobacter, Nitrococcus and other related bacteria carry out the second part of this process. This production of nitrate is particularly important because nitrate can be reduced and incorporated into organic nitrogen. And this process is known as assimilatory nitrate reduction. Now, for some microorganisms, nitrate serves as a terminal electron acceptor during anaerobic respiration, and this is a form of what's known as dissimilatory reduction. In this case, nitrate's removed from the ecosystem and returned to the atmosphere as nitrogen gas, N2. This occurs through a series of reactions that are collectively known as denitrification, and that's circled here within this cycle. This process, in which nitrate's used as an electron acceptor, usually involves heterotrophs such as Pseudomonas denitrificans. And the major products of denitrification include nitrogen gas, N2, and nitrous oxide, N2O, although nitrite also can accumulate. Nitrite is particularly concerning from an environmental standpoint because it can contribute to the formation of carcinogenic compounds such as nitrosamines. Nitrate can also be transformed back to ammonium and ammonia in a process called dissimilatory reduction. And this occurs via a variety of different bacteria, including those circled on this slide, Geobacter, Desulfovibrio, and Clostridium species. Here we have the last part of the nitrogen cycle, and this is called the anammox process. This is a recently identified form of nitrogen conversion, and the word anammox is taken from anoxic ammonium oxidation. So obviously it's an anaerobic reaction, and microorganisms that use this process use the ammonium ion as the electron donor and nitrite as the terminal electron acceptor. As a result, ammonium is reduced to nitrogen gas. So effectively, the anammox process is a shortcut to molecular or atmospheric nitrogen, N2. And this enables it to proceed directly from ammonium and nitrite without having to cycle first through nitrate. Now, what's interesting is that even though this reaction was known to be possible, microbes capable of performing this reaction were only just recently documented. So for many years, microbiologists wondered where the missing ammonium could possibly be, because calculations of mass did not agree with experimentally derived measurements of nitrogen. So the discovery that certain bacteria, marine bacteria, oxidize measurable amounts of ammonium to atmospheric nitrogen, thereby removing it from the marine ecosystem, has really prompted a re-evaluation of the nitrogen cycle. Let's now recap some of the important concepts that we've just visited. Nitrogen fixation involves the reduction of atmospheric nitrogen into various forms of organic nitrogen. This is a process that's absolutely unique to prokaryotes. Nitrogen fixation requires an expenditure of energy and a source of electrons. It's carried out by aerobes or anaerobes, and aerobes need to use a variety of, or a particular strategy to protect the nitrogenase enzyme from oxygen. The products of nitrogen fixation are immediately incorporated into organic matter as amines. Nitrification involves the deamination or removal of amine groups from amino acids. Ammonium is converted to nitrite, and nitrite is converted to nitrate. So this involves the two-step process. Let's look at the two different fates of nitrate. Nitrate may be reduced and incorporated into organic nitrogen. This is referred to as assimilatory nitrate reduction, or nitrate may serve as a terminal electron acceptor during anaerobic respiration. In this case, nitrate is removed from the ecosystem and returned to the atmosphere as nitrogen gas. This is referred to as dissimilatory nitrate reduction. Finally, we looked at the anammox process, which involves the anaerobic oxidation of ammonium using nitrite as the electron acceptor. And this results in the production of nitrogen gas. Let's turn our attention now to the phosphorus cycle. And you can probably imagine that unlike the carbon and nitrogen cycles, the phosphorus cycle has no gaseous component. And the cycling of phosphorus is still very important for a number of reasons. So for example, all living cells require phosphorus for the synthesis of nucleic acids, lipids, and even some polysaccharides. However, most phosphorus in the environment is present in very low concentrations, and it's often the growth limiting nutrient. Unlike carbon and nitrogen, which can be obtained from the atmosphere, phosphorus is derived almost solely from the weathering of phosphate containing rocks. Therefore, in soil, phosphorus exists in both inorganic and organic forms. So of course you can imagine that there's organic phosphorus found in biomass. Now the phosphorus in these organic materials is recycled by microbial activity, whereas inorganic phosphorus, being negatively charged, so it complexes very readily with cations in the environment, cations such as iron, aluminium and calcium. And these compounds are quite insoluble. So their solubility in soil solution is pH dependent. So that the phosphate is mostly available to plants and microbes between pH 6 and 7. And under these conditions, these microorganisms can rapidly convert phosphate to its organic form, so it becomes available to other organisms. The microbial transformation of phosphorus features the transformation of simple orthophosphate. And this includes the transformation of orthophosphate to more complex forms. So for example, the polyphosphates that we talked about in an earlier lecture that's found within inclusion bodies in the bacterial cell. Here we have a simple phosphorus cycle, which you've probably seen in the past. So phosphorus enters soil and water through the weathering of rocks, through the addition of phosphate fertiliser, and through surface residues of decomposition of plant and animal matter. Both plants and microorganisms rapidly convert inorganic phosphorus to its organic form, and this is called immobilisation. The phosphorus that's not converted can leach great distances or complex with cations to form relatively insoluble phosphate compounds. Microorganisms also contribute greatly to the sulfur cycle. Let's start with sulfide, S2 negative. Sulfide can serve as an electron source for various groups of microorganisms, and it's converted in this process to elemental sulfur and sulfate. Sulfate is SO4 2 negative. When this sulfate diffuses into a reduced habitat, it provides an opportunity for different groups of microbes to carry out what's called sulfate reduction. For example, when a usable organic electron donor is present, some microbes use sulfate as a terminal electron acceptor, as described here, and this occurs during anaerobic respiration. Dissimilatory sulfate reduction, that is the use of sulfate as an external electron acceptor, results in the accumulation of sulfide in the environment. In comparison, the reduction of sulfate for use in amino acid and protein biosynthesis is described as assimilatory sulfate reduction. Sulfite, SO3 2 negative, is another critical intermediate that can be reduced to sulfide by a wide variety of microorganisms. Here we have a simplified sulfur cycle. So let's start with the purple arrows at the centre of the cycle. These show that both sulfate and sulfite reductions are dissimilatory processes. Sulfate reduction can also occur in assimilatory reactions, and we can see these at the top left-hand side of the cycle. And these result in organic forms of sulfur. Elemental sulfur reduction at the centre of the cycle to the right. Elemental sulfur reduction to sulfide is carried out by various microorganisms. And the oxidation of sulfur, which we can see on the right hand side of the cycle, can be carried out by a wide range of both aerobic and anaerobic microorganisms. Finally, we have the ion cycle. And the ion cycle shown here looks relatively simple, but it's very interesting. The ion cycle mainly features the interchange of ferrous ion, Fe2+, to ferric ion, Fe3+. And we'll start with ion oxidation, which is shown here in blue. Ion oxidation can be carried out at neutral pH by certain microbial species and in acidic conditions by other species. Some microbes oxidize Fe2+ using nitrate as an electron acceptor. And these include a very interesting microorganism called dichlorosoma. And I'd just like to talk about dichlorosoma for a minute. Dichlorosoma can also use compounds called chlorate and perchlorate as electron acceptors. And because perchlorate is a major component of explosives and rocket propellants, it's actually a frequent contaminant at retired munitions facilities and military bases. So as a result, dichlorosoma may be used in the bioremediation of these sites. This process also occurs in aquatic sediments with depressed levels of oxygen, and may be another route by which large zones of oxidised iron have accumulated in environments with lower oxygen levels. Now let's turn our attention to iron reduction, shown here in red. And iron reduction occurs under anoxic conditions, and this results in the accumulation of ferrous iron. And although many microorganisms can reduce small amounts of iron during their metabolism, most iron reduction is actually carried out by specialised iron respiring microorganisms. And there's a few different species listed here for your interest. And these microorganisms can obtain energy for growth on organic matter using ferric iron as the electron acceptor. In addition to these relatively simple iron reductions, some bacteria known as magnetotactic bacteria transform iron in their immediate environment to the iron oxide called magnetite. And the chemical formula of magnetite is shown here towards the centre of this cycle, and you can see that the formula is Fe3O4. So by accumulating or transforming this iron to magnetite, these bacteria essentially construct intracellular magnetic compasses, which is quite interesting because they've taken these genes for magnetite synthesis, cloned them into other organisms, and thereby creating new magnetically sensitive microorganisms. And of course, it's important to note that in nature, cycles do not occur independently. So for example, an autotroph that fixes carbon dioxide also requires other elements for protein, nucleic acid, and phospholipid synthesis.